that I'm sharing?
Yes, sir.
Okay, so Aisha has some microphone issues. Okay, cool. So, perfect. So in the last class, what we did, Parker, would you like to just go over it quickly? You're not putting much of strain on your throat, if you can, at ease. Otherwise, I'll take care of it. Will you be able to?
Sir, I would, but I have to screen right now. Also block me.
Okay, so let's not put you through it. So I'll take care of it then. I'll give a quick summary of what we did in the last class. Okay. So basically, we talked about while we were talking of evidences of evolution, we talked about, apart from talking about homologous and analogous organs that lead to divergent and convergent evolution respectively. We also talked about adaptive radiation. We studied how the concept of adaptive radiation was identified, and it works in the case of Darwin finches in certain isolated islands of Galapagos. We also talked about adaptive radiations of marsupials in the Australian continent. And marsupials are the organisms where the young ones are born without completely, like a complete development inside the womb. So they're kind of born underdeveloped, and then that's why just after the birth, they are they reach out to these pouches in the mother's body, and that is where they develop. They have the mammary glands there because they are mammals. So they get nutrition in the pouch and protection also, and they develop to a fully mature organism. Okay, and we also talked about how multiple adaptive radiation at a particular isolated geographical area can lead to convergent evolution, right? So all these things were clear to both of you, Parker and Aisha? Yeah? Any doubts still here from the last class?
No, sir.
Okay. And then we discussed about biological evolution, what the theory tells us about, and we will go and look at some proofs and evidences for it now. So mostly based on what Darwin proposed, the biological evolution works through natural selection and branching descent. So nature selects the best trait. That's why we call survival of the fittest. And there's always a branching descent, not a linear chain where from one organism the other came and so on. But often when speciation occurs, over the course of millions of years, it has occurred in a branching descent manner, right? We also talked about Lamarck and the theory of Lamarck that was based on doctrine of desire, where that's called Lamarckism, and where Lamarck argued that the evolution of organism is driven by use and disuse of organs, right? So use and disuse of organs, he proposed that, taking giraffe as an example, he proposed that giraffe used to have shorter neck, which was true. And then he argued that the longer neck in the current giraffe population is a result of these giraffes over generations trying to stretch their neck to find food. So the beginning and the end is correct here, that giraffes' ancestors were not having longer necks, but now giraffe have longer neck than their ancestors. But the cause of it was not true, which was proposed by Lamarck, right? So Lamarck was kind of inspired by evidences that he saw around, that people can transform, or organisms in general can transform their bodies, phenotype mostly by what you call accumulating some characteristics which are called um, acquired characteristics over the lifespan. But what he was wrong about is that all these characteristics can be passed on to the next generation. Okay, acquired characteristics mostly cannot be passed on to the next generation through the genome, right? And I also kind of gave you a side, like a, a, a parallel, what do you call, a spin-off of this theory, that people now believe to some extent that many of the characteristics or traits that can't be passed on through genome, like directly changes in the genome, can be passed on through epigenetic markers, and that's the field of epigenetics. But there are acquired characters that can't be passed on even through epigenetics, right? So people are trying to revive kind of this argument for neo-Lamarckian theory, neo-Lamarckism, like we have neo-Darwinian theory, where we have made changes in these theories based on evidences that we have now. Now, in today's class, we are going to go further and talk about mechanism of evolution, and then we'll go to how animals and plants evolution happened over millions of years. So to begin with, we know now that we had many things, many answers coming from, in bits and pieces. And people were trying to put all the pieces of puzzle together to understand evolution back in time. But some questions were still open, and they needed like stitching with evidences. One of the thing is that there were two things that people were trying to wrap their heads around. One is what Mendel did, and Mendel proved that we can pass on traits and characteristics to the next generation. And there are factors, he called them factors, which are passed on. Later on, people also figured out that there are something called mutations, right? Mutations are random, directionless uh, variations, which are minor variations, but they are inheritable. They can be passed on to the next generation, but they do not happen in a directional manner for the benefit or for the betterment of an organism. Mutations are also, deleterious, or mutations are also harmful, as we know. But Darwin's theory, when he formulated the theory based on observations that he did, it looked like evolution is directional, right? For Darwin finches, if you see, it worked very well, and it worked in a proper direction. So the finches that wanted to be insectivorous became insectivorous, like they got beaks, like beaks such that it helps them become insectivorous, and so on for seed eaters and for other herbivorous, frugivorous finches. So it looks like evolution is directional, right? But mutations are directionless. And then came Hugo de Vries. Remember, this the same Hugo de Vries who rediscovered Mendel's work independently. Remember the name Hugo de Vries?
Can you please repeat?
Yeah, I'm saying, remember the name Hugo de Vries? Hugo de Vries was, I told you when I was teaching you Mendelian genetics. Hugo de Vries, along with two other people, independent. Am I audible to you, Hiram?
No, sir. Your network is unstable, I think.
Am I audible now?
Yes, sir. Yeah.
Okay, perfect. Yeah, so we're talking about mechanism of evolution, and I asked about, did you remember the person Hugo de Vries? I told you about him in during Mendelian genetics. He kind of rediscovered Mendel's work after a very long time and raised, along with two other um, um, researchers independently, and raised the work to the status of laws, right? That we now study and remember. So Hugo de Vries, based on his own work, and he worked on his model system was um, a flowering plant called evening primrose. Anyone knows the scientific name of evening primrose? It's a very famous plant in biology. Evening primrose. And based on his work, uh, he put forth the idea of mutations like that they are large differences that suddenly occur in a population. Okay. So his idea of mutations was that they are large and sudden changes that cause evolutionary shift in population. So Hugo de Vries kind of put forth this idea that mutations can lead to suddenly making of new species, right? These are these are big changes. These are not small minor changes like people talked about other factors of Mendel, or people talked about, like Darwin talked about small minor changes that lead to speciation in case of Darwin finches. He said that suddenly things can change and one species can randomly become another. But we know that mutations are random and directionless, right? But so according to Hugo de Vries, the problem was points are mutations are random and mutations are directionless, which is true about mutations. But if we think about Darwin's theory, of evolution, what does this tells? It tells us that variations are small. In this case, it was random and large. Mutations are random and large, he says, but variations are small and happens over millions of years. And when he talks about variations, and he said that variations are directional. Now, Darwin didn't speak much about Mendel's work, or maybe he knew and he kept silent because he was not able to explain it. He also couldn't comment on other things, right? So according to Darwin, evolution is gradual, right? Like it happens slow, really slow, and over a long period of time. But Hugo de Vries believed that mutations causes new species to form, and they are sudden. It's a single step large mutation event. So for Hugo de Vries theory, uh, based on these two concepts, he believed that evolution is fast and single step, and he believed that mutations causes Speciation. Now, what is speciation? Anyone knows speciation? You must have heard of speciation in your schools if this chapter is done, correct? Hiram? Parker, what is speciation? It's a simple term. What do you think it is? Just guess.
So it's how new plants and animals are formed or
Form-
created.
Yeah, formation of new species from pre-existing species, right? That's speciation. I think Aisha is also writing in the Parker is writing in the chat, more species are produced. Yes, so more species evolve from the pre-existing species. And that is very important from the pre-existing species. Species cannot just come from thin air or they cannot just produce. When we find a new species, it almost look to us like because we are finding it after millions of years now in the present. We didn't see them forming when they were in the act, right? We don't know what happened exactly behind the curtains, but we are sure that they just didn't arrive, right? So that is another thing that tells us evidence to refute the theory of creationism, that it is still happening. It's not like someone back in the past created everything. It's like a painting is created and it's there. We are just exploring it. It's like we can see evolution on in microorganisms happening live in front of us. So this whole act of new species, new organisms, new traits forming is still ongoing. So it's not a one-time job. And that is called speciation, formation of new species from pre-existing species. Exactly. So both of you are correct. And Hugo de Vries believed that mutations are the cause of speciation, and he called them as single step large mutation. Single step large mutations or large changes. And the single step large changes, the word is called saltation. Saltation. Now, saltation is a word that you will study, you will hear in biology, apart from evolution, also in neuroscience, where we call that because of the myelin sheath in the axons of the neurons, there is saltatory conduction. Remember that? Class 11th, nervous system. Myelin sheath helps in the transmission of a signal faster. Remember that? Yes, no?
Yes, sir.
So what does myelin sheath do? It makes the signal jump, right, and move faster. So that's called saltatory conduction of the signal. Here it's called saltation because, right, in one step you can make a new species, which is which had to be studied and verified. So according to Darwin, it doesn't happen like in a single uh, step, but it's more slow and directional. So to join these two, we knew that how mutations work, but we also know that all mutations, now we also know that all mutations are not favoring the existence of an organism. And only those that favor get selected, right? Not the ones that are deleterious, because the organism dies and everything, the story ends with that organism. So later when so to give an answer for these two confusions, we needed not to study just one organism or one individual. We needed some population genetics to be done. And when these population genetic studies were done, they brought more, clarity and shed light on the issue. And that one concept that made things a lot clear was given by two people, Hardy and Weinberg, and it's known as the Hardy-Weinberg principle. So what is Hardy-Weinberg principle? Like, this is very important part that it should have been covered in your schools if this chapter is done. So anyone would like to initiate the discussion? What do you understand? What is your existing understanding of Hardy-Weinberg principle? Any idea? Hiram?
I know very basic things about few things about this.
Yeah, tell me. Let's start
Uh,
with fundamental basic things. Yeah, good to go.
so it states that occurrence and consistency of a gene frequency.
Mhm.
It explains mathematically the occurrence and consistency of gene frequency for a particular gene,
Mhm.
and the allelic frequency remains constant through generation.
Okay, right. Both the sentences are correct. But what do we understand by, do you understand these sentences?
Not really.
No? Like, do you understand it? Not fully, but partially. That also will work. We can build on it. Do you understand what does it mean to be the gene frequency? You said gene frequency is consistent, right?
Yes,
And what was the other sentence?
the allelic frequency remains constant throughout generations.
Allelic frequency. So allele is basically a varied form of the same gene, as we know from chapter 5. So it's the same gene, but two different versions of the same gene. They are called alleles, right? So what you are saying is in a pool, in a population of organisms, you can mathematically find the occurrence of alleles in a population, right? Occurrence of allele of a gene, and all these alleles, the frequency in a population remains constant. So it can't change beyond a point. That this is what you're saying, right?
Yes.
So in layman's term, if I talk to you about, because we also, our aim is also to understand biology. Can you explain it using anything in our everyday life? What do we mean by consistent gene pool? Basically, Hardy-Weinberg principle talks about genetic equilibrium. You have read the term equilibrium in your chemistry, I guess, right? What is equilibrium? Equilibrium. Scientifically, what does the word equilibrium means? Come on, participate. You have to understand this before you go. So clearly, either the teachers at school are not doing a good job, or since you have a backup plan to come to me and understand, you're not paying attention in the classes. What is happening? The former or the later? Come on, equilibrium, you know. Come on. Aisha, Parker, let's not just listen to stories. Let's make concepts. These foundations will help you everywhere when you're giving exams, when you're
Science schools, they don't teach us evolution properly. They're like, you can do it by yourself. It's just like a story.
what? Why do they not teach you evolution? It's not a story. Like I think you all are convinced that it's not a story now, right? We have more than enough evidences, which I think people are sometimes not aware of or they are ignorant of. But if you don't understand evolution, I don't think you're going to understand how everything. There's a famous quotation in biology that says that nothing makes sense in biology except in the light of evolution. So though it is not too heavy a chapter from board's point of view, like you don't have to do lots of, you can just remember a few facts and go ahead and pass this chapter, but I think it's very important to understand some main concepts like Hardy-Weinberg, because they can give you a numerical on Hardy-Weinberg principle in entrances both and in the boards as well. So let's not talk about overall evolution, how bad our school systems are at teaching us. I always believe, and I always propagate it in my classes, that A person who's educated only at school is an uneducated person. So if you're only educated in your classrooms, then you are uneducated, as good as uneducated. You have to be educated out of the classroom in your everyday life, not because you want to become a scientist so that you will study science, not that because you want to become a chartered accountant so you will study maths, but because you are a human, right? You belong to a human species. Anything that you do, if you have logic in that, whether you become a chef, many people I've seen who are brilliant researchers have shifted their careers to entirely something different, which is not associated with science according to society, but they flourish, right? Just because they know logic. So education is not about making you a doctor or engineer or one or the other profession. Education is what makes you more human, more logical, and less extremist towards any kind of belief, faith, or like even science is the belief based on facts. So if you are a true scientist, you will not be extremist even in terms of saying one thing or the other. You will always seek evidences, logic, and that's how you will operate because you know how things work. So don't just ask questions. If they say that this is not important, then why it's in your textbook? If it's not important, it shouldn't be in your textbook. For that matter, many things which are important are not in your textbook or have been deleted. So I don't think even that argument makes sense. But yes, this is still there, right? Okay, cool. So Parker is writing in the chat, when both reactant and products are properly balanced. When you say balanced, it always has to be properly. There is no improper balance, Parker. So nothing can be improperly balanced. If it is balanced, it's so with respect to a chemical reaction, you're correct when both reactants and products are balanced, which means the rate at which, and it happens in a reversible reaction where the rate at which reactants are reacting to form products and the rate at which products are dissociating to form back the reactants is same and equal. At that point of time, you say that this chemical reaction is under equilibrium, right? So when you just write this one line, it doesn't make any sense. You have to say that when the rate of and the reaction is balanced, then it's called equilibrium, right? But that's good answer to begin with, Parker. Great that you remember it. So equilibrium is something where it could be applied to any process where the resultant, whether it's a directional flow of ions from inside to outside or outside to inside the membrane, anything, whether it's forestation versus deforestation rate. Equilibrium is when the net is zero. When the process is happening in two directions are equal and opposite, and the net result is zero, it's called equilibrium. There's no change. So no equilibrium means no significant change in any direction, increase or decrease, loss or gain. Make sense, everyone? That's called equilibrium. Yeah, perfect. Now this principle says, mathematically it proves that the total genes, the total number of genes, you can write down Hardy-Weinberg. There are many ways by which Hardy-Weinberg principle can be defined. So first is, write down, which talks about genetic equilibrium, the total genes and their alleles. Total genes and their alleles in a population. In a population. And in bracket, this thing, the total gene and all their alleles combined together for any population, any species, it's called the gene pool. So we have a gene pool, humans have a gene pool of their own. So will be other populations as well. So the total gene pool remains a constant. Remains a constant. And if I talk in layman's term, how many of you have seen actors look alike? Famous celebrities, they have their lookalikes. Have you seen those lookalikes?
Yes.
Yeah.
Yes, sir.
There's a common myth which is not true actually, or I shouldn't say it's not true, but to some extent you can say that a common myth in many cultures is that you have 7 people just like you in the world, who looks like you in the world. I don't know where this myth came from, but recently there has been a paper. It's a very good, extensive paper that uses artificial intelligence algorithms. They collected data from around the world, and they realized that there is a chance of someone being someone's lookalike to a great extent in the world. In 8 billion humans, there is always a chance that you will get a lookalike. It will not be 100% lookalike because genetically you both are not related. When I say genetically, like ancestrally, like the person is not from your family. So actors look like sometimes, when they have the glasses on. Why they have the glasses on and beard and contour on the face? Because they are trying to hide those subtle differences which can be easily picked in their appearance, which differentiate them um, from the actor or the celebrity. So they just kind of cover them with glasses and beard and mustache contour, but then they will do something with their hair and things, and they will look a spitting image of the celebrity. Correct? Now the question is that person related to the family of the celebrity?
No.
No, right? And how did the person got similar features, like the similar shape of the nose, the similar shape of the eyebrows, the similar face shape, the chin shape, the ear shape, the hair texture, like the celebrity? Right? So there's no magic or miracle in this. The simple answer is there is limited number of phenotypes in human gene pool that can dictate how my eyes shape and my eye color will be. It's not infinite. It's not like 8 billion different eye shapes and colors can be there in the world. It's always limited. It's always a constant pool. So basically, how human genetics work is the number of alleles for each and every gene that human has is constant. Every time it's being picked from that same constant gene. Even after some recombinations, new alleles are formed, but that's again is constant pool. And just by chance, randomly, if let's say there are 100 features, you need 20 of them which are prominent out of those 100 to be similar, to look very much alike. For example, if I open that, if I take dental samples of 2 people, they will be very, very, very different. But since we are not checking how the shape of the teeth of these 2 lookalike people are, till the time they have their mouth closed and we are just looking at the face contour, they look very similar to each other. So we say that, oh, they are lookalikes. But if we go to details, they are very, very different from each other. But since those 10, 15 features, like the shape of the eye or the shape of the chin, ear, or the face works, and these are the most prominent features, but back, many things will be very different. But these are the prominent features, so it's working well, and hence we get look-like. So this is a proof that the total genes and their alleles in a population remains constant. And that's what Hardy-Weinberg principle talked about, that it reaches genetic equilibrium for every population. Another way to define genetic equilibrium is that if I give you a population, given in a population, you can find out the frequency of occurrence of a allele, right? Mathematically, as a researcher, you can always find out what will be the frequency of this allele given that the frequency of the other allele is this. Okay. And Hardy-Weinberg principle works for a diallelic gene, like where there are 2 contrasting alleles for each gene. So write down the other way, how to define it. Hardy-Weinberg principle states that the frequency of occurrence of an allele of a gene, the frequency of occurrence of an allele or of a gene, allele of a gene, remains fixed in a population, or you can say remains constant in a population through generations. Population through generations. So it's not like it's not a fixed number for every gene, but whatever it is, let's say there can be more variations for hair texture than there can be for the shape of the nose, because shape of the nose is something very a prominent marking feature, and the structure has a function. Like we need to have a nose. Now nose cannot be something which is twisted and curled, because it doesn't make sense to have a nose like that. For example, the shape of our fingers. Now we have 5 fingers. We know that the thumb is the smallest. So in every human, the thumb will be the smallest from the other 5 fingers, and it will be exactly at the same distance from the finger, like at the same angle. It won't be much like birds. So birds have their opposing thumbs, like with which they can perch, like they can sit on the tree. Our thumbs are far enough to allow us climbing, writing, etc., and close enough so that they can still pick with all our fingers close together, right? We can bring them together, all the fingers, we can touch each finger together, and we can also make it far apart to do other things. So that's the best thing about our hand. So all humans will have things like this, but the only change will be, size, like what should the change, like the length sometimes of the overall fingers, but proportionally it will be same. But for hair, you could have more varieties. For eyes, you can have more varieties, eye color, etc. Is that clear, everyone?
Yes, sir.
A simple one-liner to state Hardy-Weinberg principle, which I like, which mostly is easy to remember and students write the most in exam, is that Hardy-Weinberg principle states that the sum total, the sum total of all the allelic frequencies in a population. Remember, it's always about population. You can't apply Hardy-Weinberg to a individual because it's equilibrium, so it has to be a population. In a population is 1. This is what it says. So the sum total of all the allelic frequencies. So all these 3 statements are saying the same thing in 3 different ways. All the 3 things are clear to you, everyone?
Yes, sir.
Now,
Yes, go ahead.
since this theory is based on mathematical principles, was it like accepted initially?
So this theory came after people had a lot of evidences that were not making sense together. So like, Mendel started using mathematics very early to explain biological phenomena. It was not accepted well in that time, but by the time of Hardy-Weinberg, people realized that mathematics can be used to explain and understand a lot of things. So Hardy-Weinberg principle, yes, because it had evidence is right there in front of people's eye, and it made sense. And what you could actually calculate it first, then go to a field and do the experiment, and you will find, oh yes, the same results come. Okay, does that answer your question?
Yes.
Yeah. So Hardy-Weinberg principle, the thing is that there are assumptions made for Hardy-Weinberg principle also, but any mathematical notion comes with some assumptions, right? Even the simplest of mathematics that you do, you say that let x be this, right? Or let the length be x, or let x be 1, and then you go ahead, right? So in Hardy-Weinberg principle also, there were some biological assumptions made. That first assumption is that for every gene, there will be minimum and maximum 2 alleles. But we know that for many genes, there are 2 alleles, 2 contrasting alleles, like for the gene for height in plants. But for the gene for height in humans, there are not just 2 genes, right? There are more than 2 genes. So there are there are polygenic inheritance as well. There are what we call as pleiotropic genes as well. So those things do not fit the best and cannot be explained through Hardy-Weinberg principle. For that, we have more higher concepts to understand. Those are also mathematical concepts. But this is the simplest one that you study at school level to understand that, yes, evolution actually is not just any story people made. It can be proven through mathematics. Evidences can be found based on what you predict also. So what this theory says, let's try to make it mathematical now. So let's start with a simple example. Tell me any diploid gene that is your favorite from Mendel's work. Let's take height, the gene of height, right? So the allele capital T will give for tall plant, right? And the allele small t will be for
What plant?
short plant or dwarf plant, correct? Now, what are the all possible combinations of this allele? So let's say
Capital
this
D.
is, yeah, this is allele 1. I'll come to that. And this is allele 2, right? This is a diallelic condition. This is where Hardy-Weinberg principle fits the best. So in a diploid organism, let's say when P plant will make gametes, only one allele will go to each gamete, correct? Yes or no?
Yes.
Yeah, go ahead. Anyone has a question? Okay. So let's say for this, the frequency of allele 1 or capital T being in a population is Q. So frequency of this allele, I am assigning it any random variable. Let's say it's Q. Or to make it easy, you can also make it A or B, does not matter, whatever you make it. So P and Q is exactly how it was done. So people just keep noting it. Even if you make it A and B, Hardy-Weinberg principle is not going to change, right? So let's say this is P, okay? And similarly, the frequency, you everyone know what is frequency? What is a frequency, people? What do we mean by frequency? The word frequency. So again, in physics, it comes from the English word frequent.
Yeah, something that occurs particular at a particular period of time.
Something that?
Something that occurs over a particular period of time.
Yes, exactly. How frequently something occurs. So frequency is with respect to, yes, Parker says how frequent wave comes at a point. Basically what she's talking about is how many waves pass through a given point, that we called that this wave is of particular frequency, 20 hertz, which means 20 waves passing through that particular point in 1 second is a wave of 20 hertz frequency. If a wave is 1 hertz frequency, it is passing only once in 1 second. Similarly, things like that. So frequency is how frequent things happen. So frequency is how frequently this allele, capital T, will be expressed in a population. From Mendel's experiment, we already have the result, right? Now let's see if mathematics also proves it. And then small t is how frequently small t occurs. I'm calling this as the frequency of this allele as Q, right? Correct. Now, simply in a diploid organism, so we are assuming that organisms are diploid. Not all organisms are diploid, but most of animals are diploid. Almost all animals are diploid. In a diploid organism, where P and Q represent the frequency of allele capital T and small t, what are the chances of an organism being tall? So capital T and capital T. For this to happen, it's this frequency. So for T, it is P, and for this T also it is P. This is the frequency. So the frequency gets multiplied. So for this to happen, the frequency is P square, right? Make sense mathematically?
Yes, sir.
Similarly, because this is pure tall, same allele happening coming from both the gametes is P square. Same allele coming from again from both the gametes is Q square. Make sense?
Yes, sir.
Yes, no? Now, if I ask you, what is the frequency of hybrids? Capital T, small t.
PQ.
One is P and another is Q, right? But mathematically, there are two ways. See, biologically, it does not matter whether capital T came from the mother and small t came from the father, or capital T came from the father and small t came from the mother, right? Because biologically, we don't say that small t and capital T. We always write capital T, small t. That's the notation, right? So biologically, it doesn't matter. But mathematically, these are two different possibilities. Do you understand that? If you understand mathematics, the another possibility is small t, capital T. Does it make sense?
Yes.
Whoever you take as the first parent can either give capital T or can either give small t. So the frequency, which is a mathematical notation, will depend on this. So mathematically, small t, capital T and capital T, small t are two different things. But biologically, it means the same. You have both the alleles, and whoever is dominant will dominate and the other will stay recessive. Do you understand this concept, everyone? So does it make sense? Yes, no, something? Yes, Parker says, can I explain again? Okay, cool. So let's forget about this for a moment. This is clear, right? If they are homozygous, capital T, it's simply from both the parents only capital T is coming. So it will be P into P, P square. Q into Q, Q square, because P is the frequency of allele capital T. Q is the frequency of allele small t. This is clear?
Yes, sir.
Now, for a hybrid to occur, one possibility is father gave capital T, mother gave small t. Another possibility is father gave small t and mother gave capital T. So mathematically, these are two different things, right? Father giving a recessive allele versus father giving a dominant allele. From mathematical point of view, mathematical frequency, these are two different things that are happening, correct? Make sense to you now?
Yes, sir.
So for one, sorry, here it will be P into Q, which is PQ, right? Here, because here it will be Q into P, which is QP. I can write it as PQ again. So basically there are two PQ, 2 into PQ, because do you understand what has been done here?
Yes, sir.
So there are two possibilities by which a hybrid can occur in a population. But biologically, we never write this notation small t and capital T. We always say that whoever gave capital T or small t does not matter. Once it is in the or offspring, capital T will be expressed. But it does matter in cases. So in biology, we always say that biology is very fascinating because it follows a rule, and you will see organisms following a rule. And then there'll be one organism that will decide not to follow the rules, because that has something to do in its evolution, where it does not follow the rule and makes a new rule. And then the lineage follow that. And then again, there is a new rule. So reptiles have 3-chambered heart, then comes crocodile. Wait, I have a 4-chambered heart. Okay, so you are exception in reptile. So whatever it becomes easy to study as exceptions, so we put them as exceptions. Similarly, always dominant alleles work is being followed, then comes some exceptions like incomplete dominance, or there is something called maternal inheritance. You know, there in snails, the shape or the coiling of their shell in snails depends on mother. What is mother sending? If mother shell was right-handed coiled, regardless of what genes father send in the next generation, it will be what mother's coiling was. So sometimes that's called maternal inheritance or maternal dependency. But you don't have to, I think it's not in your syllabus, so don't worry about it as of now. So that's where also a single rule to make or create life doesn't make a sense. So we have three possibilities: P square, Q square, and 2PQ. And what Hardy-Weinberg principle says mathematically is that the sum total of all these frequencies will always be 1. So if I write it, if I just reorient these and write it as P square plus Q square plus 2PQ is equal to 1, it will ring some bells in your head, because I can again reorient it. P square plus 2PQ plus Q square is equal to 1. Does it ring any bell?
Yes.
Much before, mathematics was much before we figured out, like Hardy-Weinberg principle was given. We know there is one equation, that is quadratic equation, a square plus 2ab plus b square, right?
Yes.
And what is the binomial? What is this? In mathematics, it is the binomial expansion of what? Yes, it is the
A plus B
binomial expression. Exactly. It is the binomial expression of P plus Q ka whole square, right? Sorry, A plus B whole square, right? Correct?
Yes, sir.
Of this. So Hardy-Weinberg principle follows a mathematical rule where it says, now, given that all these frequencies in a population are constant and stable, anything which is constant and stable can be equated as 1. So the game is like this. I have 100 balls. The total balls are 100. Now you have the freedom. There are two colored balls, green and red. Now you have the freedom to choose anything. 10 green, 90 red, 20 green, 80 red, 80 green, 20 red, whatever. But the total will always be 100. Right? It is similar kind of a situation. Now here, the total for every gene, we don't know what it is, but the theory, the Hardy-Weinberg principle says that it has to be constant. Now let's see if it is. So how to solve it is, if I already tell you that the frequency, now if it is 1, it cannot go beyond 1, right? So if there is a question that the frequency of capital T is 0.2, right? What does this mean? Or let's not say this, because it's already a good answer. I am saying that in a given population, in a population of 1,000 plants, pure tall are 300. Just this much of information. Okay, this is the question. There are 1,000 pea plants. Pure tall is just 300. Okay. Can you find how many were pure short and hybrid using Hardy-Weinberg equilibrium equation now? Hiram, Parker, Aisha, are you trying at least?
Yes, sir.
Yeah, just see if you can do it. I think I'll also have to give you something about pure short as well. But let's see. I think we need Q. It will become easier if I give Q also. So let's say short are 50. It's a number that I'm assuming. Now solve. Now I think it becomes easier, right? You have to tell me the frequency for each allele combination in the whole population. Just by knowing how many are tall and how many are short. Okay, how about if you're doing it, anyone who could solve it fast? I'll wait for a few minutes. Let's wait for a few minutes. Do it.
So we need to find the frequency of tall.
Frequency of all the allele combinations. So here, if you are taking capital T as A or P and small t as Q, you have to find the frequency for P square, Q square, and 2PQ.
Yeah, I've done it.
Sorry?
So I've done it.
Done?
For
And then
yeah.
if you have found that, you can also find for P and Q independently, right? In that population. Because you have found square, so you just have to make a root and you find that. Yeah, so what is the answer for P and Q? Hiram?
For P, is it 900?
So that's number. I'm talking about frequency. Frequency can never be more than one, no? The sum total of all allelic frequencies is 1. So if the sum total is 1, everything has to be 0 point something. Correct? I think I started with a difficult one to begin with. Okay, think about it. Think if you can solve this given information or not, and bring me the answer in the next class. By that time, I'm sure that you will be able to do it. I think by the end of today's class only, we'll see if we can do one question. Okay, but Parker says another again. Parker, you are giving me a number, and it might be correct also. But what does that number? It's just a number. For what it is the number, I'm not sure. But you have to give me frequency for every allele. So there are two alleles, P and Q. P square plus 2PQ plus Q square is equal to 1. You have to tell me what is P and what is Q. Okay.
So,
Yep.
so for P, it's 0.3.
Okay. P is for tall.
Yes.
And for Q, if it is 0.3 for P, then it has to be 0.7 for Q. Then only it can be 1, right?
Yes.
But there's a problem. You can clearly see that 300 are tall and 50 are short. So first step is to find what is not given. How many are hybrid? Now we know by biology that hybrid will always be tall in this case because there is no concept of incomplete dominance in pea plant. But how many are hybrid? 650 are hybrid, right?
Yes.
So E is present. That's where biology should match mathematics. Your biology says that 300 are tall, 650 are hybrid, which means it becomes 950. So 950 of the plants have capital T for sure. Whereas only 50 and 650, which is like 700 in total, have one or more than or one or both small t. So biologically, mathematically, it can't be 0.3 and 0.7 for P and Q. But a good way to begin. So now I have made it really easy. So think about it. Think about this question and let me know if you could do it. I'm sure you'll be able to do it if you pay some more detail and attention on it. Okay. But do you understood what Hardy-Weinberg principle is? The sum total of all allelic frequencies in a population is 1. So this is how we come to the individual frequencies of these allele pairs. And then we put it in this equation, it becomes 1. And we realize that this equation is nothing but a binomial expansion of this mathematical term that we already know of. So see if you can solve this and to just finish things in time. Let me keep it in reserve and we'll do more. I'll give you some questions to practice also as homework after today's class. And let's go forward. Is that okay, Aisha, Iram, and Parker?
Yes.
Yes,
I'll give you how to go through it. First, find you have the total numbers now. Now these total numbers sum up to 1000. So keeping 1000 as 1, which is proportionally reducing it, what will be this? If this is 1, then what will be this, and this?
0.3.
It will be 0.3, it will be 0.65, and it will be 0.05. So you can put it here, 0.3 plus 0.65 plus 0.05 is equal to 1. And then you can find what is P and what is Q. Simple. You have to solve this mathematical expansion to find what is P and Q. Make sense or not? People? Yes,
Yes, I
no?
guess so.
Okay, great. Okay, perfect. So let's, you can also do it if I don't tell you about the other. If I only tell you about P and not about Q, if I give you the frequency of P square, but I don't say anything about Q, you can still find by keeping Q as X. This is how you solve mathematics. And I just told you that the frequency of this allele is this. Can you tell me what is the frequency of other allele? Yes, you can. That's the power of Hardy-Weinberg principle. And then you go out in the field. Let's say you are studying flower color of a particular plant, and mathematically you figured out that these many plants have this number of red flowers. How many will be then white? Then what you observe in the field, you will get the same result or same answer mathematically also after putting that number. Okay, for one color. So this is how Hardy-Weinberg principle operates to say that the gene pool is constant. Now, there are some assumptions when we talk about Hardy-Weinberg equilibrium, and those assumptions, one of the assumption is that any disturbance in the genetic equilibrium, anything that can cause disturbance in the genetic equilibrium can cause a disturbance in the Hardy-Weinberg equilibrium as well. So there should be nothing happening that can change the frequency of alleles in a population. But we now know that there are some things that change the frequency of allele in a population, sometimes drastically and sometimes slowly. So there are 5 factors that affect Hardy-Weinberg equilibrium. Because it's a mathematical model, so in real nature, as I told you, that nature always is trying to evolve, find way, tussle between here and there. So it will always have some processes that disrupt something. Okay. So there are 5 factors that affect Hardy-Weinberg equilibrium. Just like in chemistry. So I remember that Parker was saying that equilibrium is reached when net reactants fusing to form products equals the total number of products that are diffusing, like dissociating to form back the reactants. That is called equilibrium. So what will happen, Parker, if I add more reactants in that beaker? Will the reaction now be in equilibrium, or it will be disturbed from its equilibrium? Disturbed, right? What will happen if I add more products to the reaction? Again, it will be disturbed. Or what happens if I add catalyst? That's an interesting question. It will again will be disturbed or not? No. Yes. Yeah. So what will happen if I add catalyst? Sorry for testing chemistry, but yeah, science is all combined.
It makes the reaction faster.
Yeah, it just increases the rate. It doesn't itself participate in the reaction. Perfect. Okay, so let's come back and talk about these 5 factors that can affect Hardy-Weinberg equilibrium. The first is very important. Genetic. So how many of you know about gene flow? So first thing is gene flow. It's also known as genetic migration. So if I allow the population to go and reproduce with another population, okay, which are separate, but we allow them to interact together, then this equilibrium will be disturbed, and it will take a very long time again, including both the populations, to reach back to, of course, over again after million of years, when these populations become very close together, there will again reach a point when there will be genetic equilibrium, but it will be disturbed at that moment, and you will not get similar results, predictable results using Hardy-Weinberg equilibrium. So gene flow is, for an example, there is a group of red beetles. These are all red beetles living in a forest. And then there are green beetles also living among them, right? So we can see that red beetles here are more in number. Now I'm making them more in number. And green beetles are less in number. Make sense? So this is one population which has its allele distributed and it's at equilibrium. But if I allow green beetles or red beetles, let's say, to migrate from this place and go to some other place because of any selection pressure to interact with other populations. So if there is a slow movement of organisms having a particular kind of a trait or allele, out, then it's called gene flow. But this happens slow and gradually, not normally in nature. It happens slow and gradually. Okay. What happens faster is called gene drift. So it's not flow, it's drift. What does the word drift means? Drift.
A sudden shift.
Yeah, a sudden change. So you might have seen racing cars drifting when a turn comes, right? So to change the direction, there are two ways for a car. So either it slows down at the turn and change the direction, or without slowing down at the same speed, it just changed the direction. So you will see the car screeching the tires and zing, and then changing the direction, right? That's called drifting. You understand what I'm saying? So
Yes.
it happens suddenly. You don't have to slow down for it. So the same word drift is used here because suddenly there will be a large scale change in a population's total gene pool. And how can that happen? Any idea? So gene flow can happen over a long time based on a selection pressure in the jungle or anything. How can genetic drift happen suddenly?
Mutation, because of mutation.
Uh, yes, to some extent you're right that if there is a mutation that is lethal to, let's say there is a mutation that happened, but then that mutation has to be induced from outside. So mutation, let's say mutation can only happen in one organism, right? If it is randomly happen, how can it happen in all the red beetles or all the green beetles? It can only happen if I start shining a UV light just to green beetles or just to red beetles and their DNA mutates, right? But again, they all will not have the same mutations. Their DNA will not break at the same point. So mutation is another factor, but you are right that mutations, even if it happens in one or two or three individuals, that can affect Hardy-Weinberg equilibrium. So
Your voice is cracked.
mutation is we'll come to mutation, genetic drift. Let me try switching off my video. Is my voice stable now?
Yes, sir.
Yes, sir.
Okay, perfect. I think it's the bandwidth issue. So let me proceed with my camera off for the next few minutes. Yeah. So mutation is again another factor that can affect Hardy-Weinberg equilibrium for sure, but it is not the reason for genetic drift. Genetic drift is something that can just happen in an environment. Independent of mutation. What is that? Let's say these are called chance events or calamities, natural calamities, right? For example, giving you an example, let's say suddenly a virus starts a pandemic, right? And there's a group of 1 million people, and this virus attacks a particular receptor in their respiratory system, very much like a COVID virus. And then all those who will have one kind of allele expressing, one kind of receptor expressing, or one kind of immune cell activation will all die because of this virus. But those whose immune system can defeat it or who have something changed will survive, right? So that's like a natural calamity which within days wiped out one set of alleles and the other set of alleles just stayed back. This will change Hardy-Weinberg equilibrium big time. Another very layman example is, let's say these beetles were living in the jungle, in the grass, the jungle floor. Suddenly there is a stampede of elephants. Elephants started, duk, 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 they all started running through that patch of the jungle where these beetles are. And just by chance, all the green beetles, or not all, 90% of the green beetles died, but only 10% of the red beetles died. Then again, there is a huge equilibrium shift, right? Yes or no?
Yeah.
And why we calling it genetic drift? It is just like gene flow only, but in gene flow, organisms were moving out or in of the population very slowly. Okay. And by their own behaviors. But genetic drift is where population organisms didn't want that or didn't know that, and it just happened suddenly by chance. Then comes mutation. Mutation is random, directionless, and of course, if a population is more susceptible to mutations, and if the mutation rate is more in for a given kind of organisms, Hardy-Weinberg equilibrium will not be present there. It will be difficult to have a equilibrium of their gene pool. So that's why most of the populations try to reach a equilibrium for mutation rate also. Like, they shouldn't be mutating too much, but they shouldn't be mutating too less or not mutating at all. So Hardy-Weinberg equilibrium does not exist for COVID virus, let's say, for now, because it's mutating a lot and mutating very fast. So you cannot say with surety, given that one kind of allele is there, what will be the frequency of other alleles occurrence. And that is the whole problem in tackling this virus, because there are new mutations, new alleles coming up, and it has yet not reached a equilibrium in its gene pool. The fourth is, we all know, what else can bring change to equilibrium in a population? Genetic recombination, right? Which happens during sexual reproduction. Yeah. Addition, yes. Parker says addition of gene. When you say addition of gene, I mean, you say that new combinations are being added, right? So genetic recombination puts new combination that none of the parent had, that we know. And this also changes Hardy-Weinberg equilibrium. Yeah. So different, I should say, I will say, Parker, that you should say different combination of genes or alleles. Okay. Yeah. Though different genes also come up by the interaction. Yeah, that's true. And anything else? What is the fifth one? What are we leaving? There should be something else. Yes, the final pressure, natural selection. If the environment is changing very rapidly or faster, if there is no stable environment, again, Hardy-Weinberg equilibrium will be affected. So in nature, what happens is, for any population, there is an equilibrium, then it's get affected either by gene flow, by gene drift, by mutation, by recombination, or natural selection. And whenever the equilibrium changes, new speciation occurs. Speciation cannot occur without disturbing the Hardy-Weinberg equilibrium. Do you understand that, everyone? For now, if we are saying that humans, all humans across the planets are just belong to one species, but it won't be the case forever, and it was not the case like forever back in past. So every time you need to do speciation events, you need to create new species, breeds, you have to disturb Hardy-Weinberg equilibrium. Make sense, everyone?
Yes.
Yeah. So the fifth thing is natural selection. So if suddenly how natural selection works in this beetle population, example is, suddenly, let's say the jungle is green. So both red and green beetles live on trees which has green leaves. So suddenly there is a predatory species of bird that came to the jungle, and they love eating beetles. Which colored beetles they will pick first and fast on a tree? Red or green?
Yeah.
Red, right? So over time, their reproductive rates are same, but over time, which population will start declining and which will start proliferating?
Green will be proliferating.
Green will start proliferating. The reason there is not gene flow, not genetic drift, not mutation, not genetic recombination, it's natural selection. So nature is selecting the fittest, which can camouflage, which is the green beetles, and the red ones will eventually decline to a level where they can even go extinct. So this is how, by studying Hardy-Weinberg equilibrium, we can also study how much we should tweak or how much we should not tweak any gene frequency in a population to prevent extinction events from happening. Okay, so we apply these kind of studies on a broader scale to make models where we study organisms which are vulnerable, endangered, how fast and how quickly they can go extinct by activities which humans are doing, which is constantly disturbing their Hardy-Weinberg equilibrium. Okay, so these 5 things, 5 factors can affect Hardy-Weinberg equilibrium. And if you want to study Hardy-Weinberg equilibrium in lab conditions, we tend to keep all these 5 constant. We do not allow. So suppose I am studying this in fruit fly, I do not allow gene flow to happen. Like my flies cannot fly out and flies from wild cannot come in and join the population. We do not allow genetic drift or mutations, or we do not have to allow genetic recombination by randomly allowing flies to mate. So I'll keep them separate. And of course, no natural selection. So they should be kept at a fixed temperature environment, a constant environment across generations. Then only we can study Hardy-Weinberg equilibrium. Okay, cool. Now, sometimes these frequencies change, as I said, because of any of these 5 factors. And when the change happens, the change is so drastic, very, very different. It's so different that a new set of population comes and becomes different species. That's called speciation. So you can write down one point. This speciation events can only occur when Hardy-Weinberg equilibrium is disturbed greatly. Speciation events only occur when Hardy-Weinberg equilibrium is disturbed greatly. And the change in allele frequency is huge or large. And the change in allele frequency is large. And when this happens, the original population from which this new population, a new species arose, then that original drifted population we call as the founder population. Write down. In this case, next line, in this case, the original drifted population, from which the new population arose, is known as the founder population, because they founded, they founded a new species. So it's known as the founder population. So it's the new population that drifted, right, from the originals, which were very, very different. Now they are so different from their ancestors that they will go on to form new species. And they will be very few in the beginning. Whenever new species are occurred, it's not like from a population of 1 million organisms, another 1 million will come and become new species. It will always be a few. The few which are very different from the original pool. For these few, the allele frequency is so drastically different that they no longer are in Hardy-Weinberg equilibrium with the population. So they are drifted ones. And these drifted organisms, which are very few, become the founders of the next population that will grow from them, right? So they become the founders for the new species or new population. And this whole effect is called as founder effect. And founder effect has been noticed and has been studied in laboratory conditions using organisms that have a very fast reproductive cycles. So yeah, this is a proven thing now. So everyone understood what is a founder effect? People? With microbes, bacteria, yeast, fungi, even lower organisms, invertebrates, many experiments prove speciation by disturbing Hardy-Weinberg equilibrium by one way or the other, and making founder populations and proving this founder effect. So is founder effect clear, everyone?
Yes, sir.
Okay. Anyone has, I think we are out of time, it's fine. But anyone has any questions?
So I have a question.
Yeah. Yes, go
So,
ahead, Hiram.
it's not related with evolution, but I have exams, so I have
Okay.
a
Yeah.
doubt in other chapter.
Okay, cool. So what I'll do is, if Aisha also wants to stay, she can stay to listen to the question. But for this particular topic, we can declare the class here, right? And we can continue discussing your doubts. Yeah. So I'll see you all in the next class, which will be the last class for this chapter. And that will be a very, very interesting and fun class, because I'll be giving you evidences for human evolution at the very end. And that topic is not a lot asked in entrances, sometimes asked in boards, the sequence in which human species came up. But yeah, but I'll teach you three things: evolution of plants, animals in general, and human evolution in the next and the last class. Yeah, go ahead, Iram, with your questions.
So in human health and disease, in the cancer part, we've got two types of cancers.
Sorry. Go ahead again. Go ahead again. Yeah.
In the cancer part, we have two types of tumors: benign tumor and malignant tumor, right?
Right.
So malignant tumor is a tumor that kind of passes, invades and damage the surrounding normal tissues.
Right.
So per se, if that tumor passes to our brain and we have a tumor in our brain cells, and
Right.
if through chemotherapy you get it out, so any memory that resides on that part will also be perished out.
So if I understand your question correctly, you are saying two things. So let's say, so you chose brain. Let's not choose brain, because brain can protect itself way better than other tissues, because brain has a blood-brain barrier of its own. But if I understand your question, it means that, let's say there is a cancerous tumor anywhere in one part of the body, and it moves to another part of the body, right? Let's say from liver to
Yes.
lungs, or from lungs to any other part.
Yes.
Now you are asking is, by giving chemotherapy, you said, we can get rid of that tumor, or we can get rid of that cancer. Will there be any memory left, is what your question is, right?
Yes.
Yeah. Yeah. So first, the thing is, we have to understand that what chemotherapy does. So chemotherapy does not take out the tumor. You can only take out a tumor through surgeries, but in a cancer, often all these approaches, chemotherapy, radiotherapy, and surgery, is done either together or in combinations. So what you do with chemotherapy is you send chemicals in the body that, in a targeted manner, should kill the cancerous cells, right? But chemotherapy alone does not work 100% of the times, and also it has some side effects that, apart from killing, how does it identify as a cancerous cell, is that they are rapidly dividing cells, right? That's one property how we can find cancerous cells in the body. You might also know that people who undergo chemotherapy lose all the hairs on their body, correct? That's a side
Yes.
effect of chemotherapy. Do you know why that happens? Any idea with the hint I gave you? because
targets your
yeah, sorry, kind of.
cells, your hair cells.
So these drugs will also kill your hair follicle cells, right? Because they are also rapidly dividing cells. We continuously keep growing hair all the time, doesn't it? So it that's a side effect, right? That's how it kills. So it also kills some off-target non-cancerous cells, right, in the body. So by chemotherapy, we cannot get rid of cancer all the time completely, and that's why we if the tumor is big, what we first do is we surgically remove the major chunk. Let's say 90% of the chunk we have removed, but we are still suspicious that some few cells here and there which are in the lymphatic system, which are already malignant and are moving from one part to another, we are not aware of. To kill them, we then do chemotherapy and radiotherapy targetedly. And sometimes, if all these three done in a proper time, at an early stage, the person can defeat cancer and comes out of it, and goes into a period where still some treatments continue, and then it stops once we are sure that there's no malignant cell left in the body to cause cancer again. But always, even if one or few cells which are malignant are left in the body, and if they are in the lymphatic system through which they can spread to other part, you don't need too many cells or kind of a memory. Only one cell will go and seed somewhere else. It's like a seed, a spore, and will start dividing and forming another cancerous tumor there. So it's always a possibility that it stays if you're not able to kill all.
So,
Right? Yeah.
So like this chemotherapy will target all the cells that are rapidly dividing. So we have
Yes.
got cells that are rapidly dividing in our intestine. So those cells will have an impact?
So that's a good question. I'm not very sure if so. There are two kind of rapid divisions. One is for repair, and one is for growth, kind of like regeneration. I'm not sure. I'm sure that to some extent, people who are undergoing chemotherapy will be affected with their repair rate as well. But I'm not sure if every place wherever we have rapidly dividing cells they will be affected. But the fact that hair follicles are affected kind of give us a cue or a clue that it's possible, right? But I had to look at it to be able to say it with surety that yes, the cells of intestines are. I understand what you are saying, that there are cells in our whole throughout our digestive systems which keeps on dying and then repairing, right? Because of food continuously passing through it. Yeah, that's a very good question actually. I'll figure it. I'll see if I can find some evidences to give you a proper answer with evidence. But my informed guess is yes, they might be getting affected, but not to an extent where your survival is at stake, because your survival is anyways is at stake if you have a cancer in the body, right? So if given a choice between getting rid of cancer versus not getting your hair follicles or some cells repair and grow, we will always choose the first, right? Maybe.
Yes.
Yeah.